So anyway, I think the last thing I was saying with regard to the last teaching time is the major difference between prokaryotic, eukaryotic. Prokaryotic cells no nuclear envelopes, so the entire process can happen basically spontaneous at the same time, simultaneously. You can have transcription and translation, and actually the polypeptide being formed as the RNA is being transcripted. But in the eukaryotic cell, the transcription takes place in the nucleus, and then there's some modification to the RNA, the messenger RNA, which we will look at. So changing and looking for possible problems and fixing them before that messenger RNA leaves the nucleus and then finds a ribosome, where translation can take place, and then even with that polypeptide that's made, there can be some alterations to it. So quite a bit of difference between prokaryotic and eukaryotic cells. The primary transcript is the initial RNA transcript from gene to processing. So the central dogma, which is a term that is used, or the main thing with regard to genetics, is that cells are governed by a chain of command, DNA, RNA to protein. And that's the whole idea behind this. It sounds very grandiose. And it sounds very ho-hum to us now because we accept it, we know it, it has been highly studied, but they didn't know. This was very, very earth-shattering to figure this out and find this out. And so it became one of the overarching main ideas with regard to biology, is that DNA, which is inherited, we've talked about this from the very beginning of the class, this inherited capacity. Now we have put it together into how does that DNA work to dictate what you will look like, how you will manifest, how your body will work, how you will grow, etc. So how does that DNA actually instruct the development of any species? Then talking a little bit more about, going a little bit more about into this transcription and translation. The thing that we need to explain to you now, or gets explained to you now, is why it's a triplet code. So the DNA eventually dictates proteins. They have discovered 20 amino acids. Amino acids get together in various different sequences to form proteins. But there has to be the capacity for DNA to design these 20 amino acids. We know there are 4 nucleotide bases, and if in fact the nucleotide bases got together in just groups of 2s, you wouldn't have enough different groups of 2s to make 20 amino acids. You'd only have, I believe your book says 16 different combinations. But if in fact the nucleotide bases get together in groups of 3s, then you have 64 different combinations. Plenty of combinations to make the 20 amino acids, plus you have some overlap. And so the idea is this triplet code with regard to DNA and groups of 3 nucleotide bases, that's what we're talking about with this this triplet code, is the simplest that you can get the design and still have enough flexibility to make all of the combinations needed. So it was a combination of wanting to be very simple, this is assumed, but also have adequate variety to do everything that needs to be done. And this triplet code series, so the flow of information from gene to protein is based on this triplet code. and this will be the way we talk about it over and over again. That triplet code is actually called a codon, and what you see is, if you look at the image, the DNA strand, acclimate yourself, 3,5, there you have an ACC. The RNA complement to that's going to be UGG, and we know that the UGG triplet codon codes for the amino acid tryptophan. And so what happens is each of those 20 amino acids corresponds to a grouping of 3 nucleotide bases, which is called a codon, and that then can cause the variety that's needed for the 20 amino acids, and then remember, those amino acids can be strung in different varieties too, to make infinite trillions of proteins that are needed by living organisms. Then the words then are translated into a chain of amino acids that then forms the polypeptide. So the only thing we've really introduced here is this idea of a codon, so that you understand that the codon is a group of 3 there are 3 nucleotide bases. And then what happens is, in the transcription, one of the 2 DNA strands, called the template strand, is going to create the template for the creation of the RNA, so the complementary nucleotides for the RNA. The template strand is always the same for a given gene, and during translation, messenger RNA-based triplets called codons are read from the 5' to the 3' direction, are read that specific way. It's very important that it gets read that way, because we are using these nucleotide bases over and over again. And what you want to think about is like reading a sentence. Each codon represents a word. If you don't read the sentence in the correct order of words, it's not going to make any sense to you. And many times when this gets taught, it gets taught it's not the same as learning words, but it gets taught that way so that you get this idea that this is the way, this is the regularity, or this is the pattern that needs to take place. And so what will happen, and you will become a geneticist this way, or an expert this way, each codon specifies a particular amino acid. And there are very, very useful charts. This happens to be one. This happens to be one that we use in lab, and we also use in lecture testing situations, I guess you might say. So each codon specifies amino acid. It's placed along the position with regard to the polypeptide that then is reflected in the DNA. This polypeptide is reflected in the DNA, and as a matter of fact, you can take a polypeptide and actually make some assumptions as to what the DNA was coding for, so what the nucleotide bases were. All of 64 were deciphered by the mid-60s into this this graphic you have here, which this graphic shows up in a couple of different ways. Don't get confused. It does show up in a couple of different ways. 61 of the codes are for amino acids. What you have is some redundancy. We didn't suddenly get confused here. So you have some redundancy with regard to some codons coding for the same amino acid, but you don't have any lack of specificity. So there is, so you'd have maybe 2 triplets coding for one particular amino acid, but they always code for that amino acid. There's also 3 triplets that are stop signals, that this is the time to stop this particular polypeptide, and then you have an initiation code also. So you have a start and a stop, plus some what do I want to say? Some that are redundant. More than one codon may specify a particular amino acid. They have to be read in the correct frame in order to create the polypeptide. We will use this in lab. We also will use this in lecture. But all you're doing is you are taking the messenger RNA unit that you have been given or that you have figured out from the DNA and looking to see what amino acid does that correspond to, and you are making your nice polypeptide chain. The interesting thing about this after it was discovered, I guess it's interesting, but it's also where some of the ethical questions come in, is genes can be transcribed or translated, and after that happens, they can be transplanted from one species to another, which is interesting. They can be spliced in. So you have some, what you have here, these are both from your book, is we have made fluorescent fish, I guess that's a thing, and then fluorescent plants. So it's it can become an ethical question there. So you could, if you are more of an ethics or a humanities student, you could stop and think, oh, I don't know if I want that to happen or not at this point. But keep going on with the chapter. In terms of transcription, again, the first stage, RNA synthesis there's an enzyme that actually does the job. It's called RNA polymerase. Same RNA polymerase that we're familiar with regard to the actual replication of the that was DNA polymerase. This is RNA polymerase, so similar type of enzyme. It pries the DNA strands apart. Actually, I guess it is the same thing, RNA polymerase. It isn't DNA polymerase. I'm sorry, I got confused. Same, prying the DNA apart and hooks the RNA nucleotides in. So you have the kind of the same process where you have a bubble, you have that promoter, you have that bubble and prying it apart, and then RNA coming in. The only difference, actually it's very similar to what you learned about how DNA is replicated, but the only difference is there's a uracil substitute for thymine. So every place you had a thymine in DNA, you have a uracil in RNA. That's really the only real difference. You still have the elongation, you still have the initiation, the elongation, and the termination. So if you did a good job of learning how DNA is replicated, this synthesizing messenger RNA is very simple. So you kind of have that down, which is good. And then what you have is this building of the RNA, and then you have a RNA transcripted that actually is hanging off there in step 3, the termination. That then will be taken off, and then the DNA can get back together again, and the RNA polymerase can go and do some other job for itself. The 3 stages, as I said, elongation, initiation, elongation, and termination, starts with a promoter signal, and then several dozen pairs start from that point upstream. The factors mediate the binding of RNA polymerase and the initiation of the transcript. Basically, then the process moves along. There is a promoter area of nucleotide bases that are TA. Some people call it the Tata box. It's the crucial in forming the initial complex. So anytime that TA sequence is seen, that is thought of as the beginning. That's the beginning, the starting point with regard to the RNA, being created.